Hi everyone, my name is Abby Fister and I am a senior in the MSTC program at Paul Laurence Dunbar High School. Today I will be presenting my capstone research project on the epigenetic mechanisms of inorganic arsenic-induced cancer development. This project was conducted under the mentorship of Dr. Yvonne von Duffe-Middendorf and Meredith Eckstein in the Department of Molecular and Cellular Biochemistry at the University of Kentucky. I will begin with a brief overview of what this presentation will cover. First, I will go over some basic background information in order to aid in understanding my project. Next, I will state the research question and the hypothesis I proposed. Then, I will discuss the methodology used throughout my experiments. Next, I will share the results I collected as well as conclusions I was able to draw from the data I gathered. Finally, I will share areas for further research, present my acknowledgements and references, and then I will be happy to answer any questions you may have. Millions of people around the world are chronically exposed to low levels of inorganic arsenic. This exposure can be either from contaminated drinking water, certain foods, or industrial occupations such as coal mining. Long-term exposure to arsenic can lead to many devastating health effects, including cardiovascular disease and some forms of cancer. These issues are especially prevalent in the Appalachian regions, where coal mining often contaminates the groundwater with heavy metals such as arsenic. However, it's not really well known how arsenic exposure can lead to cancer, so future studies in this area will hopefully aid in the improvement of future cancer treatments. Past studies suggest that arsenic exposure may affect DNA through epigenetic changes. This means that the arsenic does not affect the DNA sequence itself. Instead, it solely affects how the DNA is expressed. In this case, arsenic is affecting the compaction of chromatin, which consists of repeating units of nucleosomes, shown in the figure on the right. Each nucleosome is comprised of a strand of DNA wrapped around a set of proteins called a histone octamer. Each histone octamer is comprised of two sets of 4 different types of proteins, noted as H2A, H2B, H3, and H4. At different times throughout the cell cycle, proteins called histone variants can exchange with these 4 main proteins in order to affect the compaction of the chromatin. Now, compact DNA cannot be expressed as easily, since proteins related to gene transcription and translation cannot access that tightly bound chromatin and the DNA within that chromatin. So arsenic is increasing the rate at which these histone variant mutations occur, and sometimes this can increase chromatin compaction and therefore decrease gene expression. Other times it can have the reverse effect and decrease chromatin compaction and increase the gene expression. However, in my project, I am mainly focusing on H2B variants, which have been found to tend to increase chromatin compaction, which means that it decreases the gene expression. Now for my project, the goal was to determine how the rates of certain histone variants were affected as arsenic exposure increased. And I chose to focus on the H2BH, H2BN, H2B, and H4 histones. And I hypothesized that as arsenic exposure increased, these histone variant levels would increase in the more compact chromatin, which would lead to an overall more compact chromatin structure. And like I mentioned earlier, this would result in a reduction in gene expression because the chromatin is so tightly bound together. First, I had to perform a series of salt fractionations in order to separate my protein samples into the different levels of compaction. And this was done by adding buffers of increasing salt concentrations in order to isolate and extract the different types of chromatin compactions within my samples. And after separating these types of compactions, I was then able to use those samples in future Western blots. The different types of chromatin compaction levels can be shown in the bottom row, where nuc is the least compact chromatin in the cell, and pellet is the most compact chromatin. Here is a table describing my 6 different treatment groups. While I was not able to be involved in treating the cells with arsenic due to safety precautions, I was able to perform salt fractionations for each of these different types of proteins after they were exposed to arsenic. First, I had samples of cells that were non-treated with arsenic, and this served as my control. Next, I had protein samples that were exposed to arsenic for either 3 weeks, 8 weeks, or 17 weeks at a 0.5 micromolar dosage. Then I had samples of cells that were exposed for 26 weeks at a 2.0 micromolar dosage. And finally, I had my reverse-treated cells, which were exposed at a 2.0 micromolar concentration for 26 weeks, and then the arsenic was taken away for an additional 3 weeks in order to see if arsenic's effects could be reversed after a short recovery period. After collecting all of those samples using salt fractionation, I was then able to use them in a series of Western blots, which was done to detect the presence of specific histone variant proteins in these samples. First, I would make a 15% SDS polyacrylamide gel and run my samples of protein through that gel using gel electrophoresis at 150 volts. Next, I would transfer the proteins in that gel to a more durable membrane that could later be imaged more easily. This membrane was then blocked with a solution that would prepare it for the incubation of future antibodies. So next, I would then probe that membrane with antibodies specific to the histone of interest. And finally, after all of the antibodies have been added and removed with PBST, I was then able to image that blot using a Typhoon machine. Now for my experimentation steps for phase 1 of my experiment. During these Western blots, all of the samples on each blot had the same amount of arsenic exposure. However, the protein was separated into the different levels of compaction using salt fractionation. And this was done to compare the rate of histone variant exchange with the different types of chromatin compaction. And this process was done for the H2BH, H2BN, H2B, and H4 histones. After I performed phase 1, this led me to go into phase 2 of my experiment, in which I reversed the orientation of all of my Western blots. So now, all of the samples in each Western blot had the same amount of compaction, however, they were separated into the different types of arsenic treatments. And this was done to compare the rate of histone variant exchange and the different arsenic treatments. And this type of Western blot was performed for the H2BH and the H2BN histones. And here are the Western blots I collected for phase 1 of my experiment. On the top, you can see the different types of chromatin compaction, where nuke is the least compact chromatin in the cell, and pellet is the most compact. On the left, you can see the different arsenic treatments from non-treated down to reverse-treated, and you can also see that all of the histones I found had a molecular weight between 11 and 17 kilodaltons. And here from left to right are the Western blots for the H2BH, H2BN, H2B, and H4 histones. A band indicates that the histone was present for that particular sample. Now, in the soup fraction, which is low compact, for each of these blots, you can see that a band typically is present for the non-treated week 3 and week 8 arsenic exposures. However, as the arsenic exposure continues to increase, this soup band tends to disappear, meaning that the histone is no longer present in that low compact chromatin as arsenic exposure increased. Furthermore, there was a general trend that as arsenic exposure increased, the band in the compact 600 millimolar fraction, that band tended to increase in amount, meaning that the histone was present in the more compact chromatin as arsenic exposure increased. However, it was a bit difficult to make these qualitative comparisons between the different blots, so that's what led me to go into phase 2 of my experiment, in which I reversed the orientation of the blots. And now you can see that the bands in the nuke fraction tended to decrease in amount as arsenic exposure increased. However, there was a very heavy band in the reverse-treated fraction, which was not expected. And on the bottom, you can see that the the histone variant was present in relatively equal amounts for the different arsenic exposures. However, there was not much of the histone variant present in the week 17 chromatin. So looking at this data, overall, it did support my hypothesis in that as arsenic exposure increased, the histone was present more frequently in the more compact chromatin. However, looking at the heavy band in the reverse-treated fraction, it can also be noted that arsenic's effects could not be reversed after a short recovery period, since the non-treated and the reverse-treated bands did not match up equally. A possible explanation for this very heavy reverse-treated band could be that after so many weeks of arsenic exposure, the chromatin became sort of accustomed to the arsenic being there, and after it being taken away so suddenly, the DNA became very dysregulated and unorganized and simply loosened up after so many weeks with the arsenic being there. However, I will need to repeat that experiment in order to make sure that this wasn't simply a mistake when I was adding the protein. And furthermore, like I mentioned earlier, with the chromatin being more compact, that leads to a reduction in gene expression because the chromatin is not accessible for proteins related to gene transcription and translation. And this is especially detrimental when considering genes that code for proteins that regulate cell division, because without the production of these regulatory proteins, uncontrolled cell division or cancer could eventually occur. And this project simply looked at identifying specific histone variants involved in the process, so that in the future, studies could be conducted that really focus on these histone variants and try to determine how they're able to produce those effects and why they end up leading to cancer. Now, there were various sources of error throughout my project. One of these sources could be from accidentally misloading protein into the gel, which, like I just mentioned, it could possibly be their explanation for why there was that heavy band in the reverse treated fraction on the reversed blots. So in the future, I would need to repeat this experiment in order to make sure that this wasn't a mistake. Also, it was a bit difficult to make qualitative comparisons between the different bands. So in the future, I would like to investigate quantitative methods to assign quantities to each of the bands so that I could test for statistical significance and draw more accurate conclusions. Now, for future research, I would first like to extend phase 2 to encompass all of the different arsenic treatments and compactions, because right now there are a few different ones missing. I could also test for different types of histone variants in order to see if this trend continues. I simply selected these 4 histone variants due to the availability in my lab. However, there is a wide variety of histone variants that I could have chosen, and I'd like to investigate those in the future. And finally, I could either lengthen or shorten the durations of the different arsenic exposure treatments. Specifically, I would like to target the recovery period of the reverse-treated proteins in order to see that if arsenic could be reversed after a longer recovery period. So in the future, I would like to extend that recovery period to see if that could happen. Now, I would like to take a brief moment to thank all of the wonderful people who helped me throughout my journey. First, I would like to thank the Math Science Technology Center and the Kentucky Young Researchers Program for giving me the opportunity to conduct this research as a high school student. Next, I would like to thank Dr. Yvonne von Dufe-Mittendorf and Meredith Eckstein for all of their mentorship and guidance in the lab. And finally, I would like to thank friends and family for all of their support throughout this long process. Here are my cited references, and I would be happy to answer any questions that you may have. Thank you for listening.